doing problem number 1. This is our diagram of DNA replication. In our first picture, we have a short little picture just showing you the lagging and leading strands. The important stuff, the second picture, we're going to start off with our DNA helicase. This is what actually goes through and splits the DNA apart. The single strand binding proteins help keep the DNA from closing back up on each other. Right here we have our primase, which puts down an RNA primer. And then right here we have our DNA polymerase 3. That actually comes through and synthesizes the new strand of DNA. Our DNA polymerase 1 will come through after our DNA polymerase 3 and fill in the different gaps that are left behind. And our DNA ligase also helps the DNA not close on each other.
Well, it reconnects them.
It reconnects them, and that's actually it. So.
All right, so I was in charge of number 2 and number 3. So for 2, two is our definition. So for A, it has this protein complex synthesizes primers in eukaryotes. We've got the primosome. For B, it says the protein promotes the separation of the AT-rich sequences within the ORIC, and that is DNA A. For C, it says the subunit of DNA polymerase 3 that contains proofreading activities is the epsilon subunit, or E. For D, this protein synthesizes the leading strand in eukaryotes, and that is the DNA polymerase epsilon subunit. Right? Okay. Just want to make sure I hadn't written it down wrong. All right. For E, we've got, oh my gosh, I'm going to butch this name. Hold on one second. I wrote down TUS, but I know that's not what it is in its actual term.
Termination utilization substances.
Okay, so for E, we have termination utilization substances are the protein that displaces the DNA polymerase 3 from the chromosome to terminate replication. The protein that has a 5'3" polymerase and a 5'3" exonuclease activity is DNA polymerase 1. G says the protein introduces negative supercoils into the DNA ahead of the replication fork, and that is DNA gyrase. For H, we have the origin recognition complex, which is the protein that binds directly to our ARS elements. And our single-stranded binding proteins are the protein that prevents DNA strands from annealing during replication. DNA ligase is the enzyme that seals those Ozaki fragments into a continuous strand. But yeah, it says polymerase 1 does that. Anyway. Okay, so for 3, it says list 5 enzymes that contain or that consume ATP during DNA replication. We've got helicase, gyrase, primase, ligase, and DNA A. There's supposed to be a DNA in front of all of these. I just got lazy and didn't want to write it. For B, it says list 5 enzymes that consume DATP during DNA replication. We've got polymerase 1, polymerase 2, and then all the subunits of polymerase 3, alpha, beta, and epsilon. For C, it says list 5 enzymes that have 3 to 5 exonuclease activity. We've got polymerase 3, and then the subunits, alpha, beta, and epsilon. We were kind of questioning whether it was primase in the pCNA. It just made sense, so we decided that those were the other 2 options. For D, it indicate one enzyme involved in replication that uses DNA as a template to synthesize RNA. That is primase. For E, we have polymerase 3 is the enzyme that uses RNA as a template to synthesize DNA.
Awesome. All right, so number 4, I'm in charge of that this week. If you don't understand me, go and watch the teacher's video on the announcements page, because she goes through this problem step by step, and it's pretty simple. So we're given our DNA sequence up here, and we're also given our primer. So the primer follows a specific sequence in the sheet. It's written backwards, but in order for it to line up with our DNA sequence, you have to write it this way, from 5' to 3'. So then we're given these different sets of nucleotides, and then we have to figure out the different sequences that go with those nucleotides. So with A, we're given all of these, and they all just look like a bunch of abbreviations, but it's really just saying these are a dioxyadenosine triphosphate, which is what's used in the backbone of and the bases of our DNA. And since these are all triphosphates, we'll just start at the end of our primer, and remember, we're not supposed to write the primer. So you just start here with T and go from here all the way down, and that's what you get. With B, our DCMP is actually a monophosphate, so we actually can't use that because it's technically not going to exist there. So when we come up here, we'll just keep going until we hit G, because G can't exist because this is a monophosphate and C pairs with G. We just go until we hit A, and since the next one is G, that's what we get. C, same thing, we get UTP, which urethine, can't remember how you say it, is actually an RNA. So we won't actually use that at all. As soon as we hit T, then you stop, so you just keep going until you hit T. D is pretty much the same thing, except once you hit C, or something like that. Yes, then you stop. So that's that. Like I said, go watch her video. It'll make more sense than whatever I just explained. So 5, we've got all these different terms. I'm supposed to figure out which ones go with which grouping. So our first group is proteins and prokaryotes, which are like your bacteria and things. And this is what we got. We got DNA gyrase, primase, polymerase 1, polymerase 3, alpha subunit, and then 2 DNA. Those would all be found in a prokaryote. And then your mus musculus, which is your house mouse. It's an organism. It's going to be a eukaryote. You got your CDC6, MCM helicase, primase, pCNA, polymerase alpha, polymerase epsilon, and telomerase. For C, we're looking for proteins that deal with chromosome compaction. The only thing I could find was the telomerase, but again, it's kind of confusing with some of them. But D is with initiation, so we've got DNA A, CDC6, MCM helicase, and our terminations would be with TUS and your gyrase. Yep, that's fine.